Hi, today we are going to study why cells are so small. We are going to do this by investigating the relationship between cell size and the rate of diffusion. An example could make it more clear why we selected diffusion to answer this question. Well, our cells metabolize amino acids every day, and as they do so, they produce ammonia, a very highly toxic compound. Ability to get rid of this ammonia by diffusion at a higher rate is a great selective advantage. Cells that has this ability will survive longer, and produce more offsprings. And this trait will be more frequently found in the populations. We now know that cells are small because they are more efficient at transporting matter across membrane than larger cells. This is because surface area to volume ratio of a cell gets higher as the cell size decreases. That means a small cell has more membrane area for transportation comparing to a larger cell. That makes small cells more efficient at transporting matter. I will now go ahead and introduce the materials needed for this experiment. First is agar cubes as model cells of the test. You will need 3 of them with 10% sodium chloride in it. This is the whole agar cube that will represent the largest cell. This is the agar cube divided into 8 pieces. And this is the agar cube divided into 64 pieces. Identical cells, model cells, with 10% salt solutions in them. Second, a beaker of water with distilled water of 300 milliliters as the environment of the cell. Third is conductivity sensor to measure the conductivity of the solution in the beaker as a relative measure of amount of sodium chloride ions that diffuse out of the cells into the water. An interface to connect your sensor into computer for data collection. A computer with Logger Pro data collection software. Ring stand and clamp to support and secure the sensor during data collection. Magnetic stirrer and a stir bar to stir the solution in the beaker, or use a glass stirring rod. A wash bottle to clean up your conductivity sensor in between experiments. Another beaker to use as a waste container. First, connect your Go-Link interface into the USB port of your computer. You must see a green light on your Go-Link interface. After you connect your Go-Link interface into your computer, you connect your conductivity sensor into your Go-Link interface. As soon as you do this, your software will recognize your conductivity sensor. You must see it here. And if it doesn't, there's a problem. You will see a live measurement of conductivity down here. After you connect your conductivity sensor into computer, clean up the sensor piece using distilled water in the wash bottle into a waste container. And then blot it dry using a piece of paper towel. After cleaning your conductivity sensor, secure it using the clamp on your ring stand. Then place your beaker with water on top of magnetic stirrer and place the stir bar inside the beaker. Submerge the conductivity sensor in distilled water in the beaker by lowering your clamp. After you submerge your conductivity sensor into the distilled water, you go to Experiment tab on Logger Pro, go down where you see 0 and click on it. This will set it to 0. This will set your measurement to 0. After you set your beaker and conductivity sensor here, you place your model cell into the beaker, and as soon as you do this, you start data collection by hitting this collect button. Now here is our model cell, goes into the beaker, and then I start data collection and I start stirring this at a moderate speed. Data collection will stop automatically when you reach 2 minutes. Now the data collection is complete. I will stop stir. I will remove conductivity sensor. I will clean it up using distilled water in order to prevent contamination between samples. And then I'll secure it on the ring stand using the clamp again. I will dump the water and the agar cube inside the beaker, clean up the beaker, and fill it with 300 milliliters of distilled water again for the next part of the experiment. After the first part of the experiment, you clean up the beaker, change the water inside the beaker using a clean 300 milliliters of distilled water, and you place your beaker on top of magnetic stirrer again. You place the clean magnetic bar inside your beaker. You lower your conductivity sensor and submerge it inside the distilled water again. You go ahead to Experiment tab and zero your sensor again. Now you can put your model cell that is divided into 8 pieces into your beaker. As soon as you do this, however, you must start data collection. Now I place the model cell that is divided into 8 pieces in a beaker and then start data collection. Magnetic stirrer is spinning at a moderate speed again. When you start data collection, the software will ask you if you want to store the latest run, and you want to store your latest run to make comparisons between your experiments. Now data collection is complete. I will stop the magnetic stir. I will remove the conductivity sensor and again clean it using distilled water in order to prevent contamination between samples. I will remove my beaker to clean it up. I will dump all the water and the agar pieces, and I will install my conductivity sensor back onto the ring stand using the clamp. After we clean up the beaker, I replace the water inside it with 300 ml of distilled water. I place it on top of magnetic stir again. I place my magnetic stir bar inside it, and then I lower conductivity sensor to submerge it under distilled water. I again go ahead and click on Experiment tab and zero my sensor first, and then I will place my model cell that is divided into 64 pieces and set magnetic stir to a moderate speed. After I add my cells, I will start data collection. It will ask me if I want to store the latest runs. I will click yes, I store. Now data collection is complete. It's time for cleanup. You thoroughly clean your conductivity sensor using distilled water. Blot it dry using a piece of paper towel and store it in its box for longer use. As it is seen in the graph generated by the software, trends are pretty clear. But in order to make more clear picture of the results, it is necessary to further process the data collected. Since the explanation for the question, why are cells so small, is having a greater surface area to volume ratio would yield higher rates of diffusion, producing more efficient cells. Then we have to find out the surface area to volume ratio of our cells and then compare it to their rates of diffusion in order to support or refute this explanation. We compare 3 model cells that are agar cubes, a whole piece agar cube divided into 8 pieces, and an agar cube divided into 64 pieces. A side of this cube in the whole agar cube was 5 centimeters. One side of the cube that is divided into 8 pieces was 2.5 centimeters. And the cube divided into 64 pieces, the side of that cube was 1.25 centimeters. If you do the math and calculate the surface areas, and volumes are the same for each of the cube because they're identical cubes, the surface area to volume ratio would be 1.2 for the whole cube, would be 2.4 for the cube that is divided into 8 pieces, and would be 4.8 for the cube that is divided into 64 pieces. Now we must compare these surface area to volume ratios to the rate of diffusion of these cells in order to see if there is any correlation between the surface area to volume ratio and the rate of diffusion of these cells. When you look at the graph, you see 3 different lines with different colors. The line with the red color is our first run. You see its data table here, and it was our first run with the whole cube. The line with the blue color was our second run, where you see its data table here, and it was with the cube divided into 8 pieces. And line with the green color was our latest run, where you had cubes with 64 pieces, and this is the data collected in the table. Now we're going to create another page and plot the data in the second page. I will gather data for it from here and from our calculations. From this page, I will gather data that is the diffusion rate for each of the cubes. How am I going to do that? Well, I'm going to select an area of this graph where it is mostly linear. So I assume that after the 50-second, the entire graph is more or less linear. So I will select this side of the data by just clicking on it and dragging my mouse over the graph. After selecting this portion, I will go and click on this button where it says linear fit. That will produce a best fit line for all of the data collections and will do some statistical analysis for me, including calculating the slope of the line and producing a linear fit equation for them. When I click on this, it will ask me for which runs do I want to perform the linear fit, and I will select all of them because I'm going to use this data to create a graph. Now these are floating labels. This is for the latest run, this is for the second run, and this is for the first run. And it shows me the slope, which is the rate of diffusion. Slope of the first run, rate of diffusion of the first run. That shows me this slope, the rate of diffusion for the second run, and that shows me the rate of diffusion for the latest run. Now, since I already have the rate of diffusion for each of the runs, I will go ahead and create a new page and create a data table for surface area to volume ratio versus the rate of diffusion. I click on this tab where it says page, and I click on add page. I will click on new data set and graph, and I will hit OK. In a data set, on my x-axis, this is the independent variable, and in my experiment, what I change, what I manipulate, which is the independent variable, is the surface area to volume ratio of the model cells. And it was 1.2 for the largest cell. It was, the surface area to volume ratio was 2.4 for the medium cell, and 4.8 for the smallest cell. And now I go back to the first page and see what's the rates of diffusion for these runs. I switch between pages by going into this drop-down menu. I click on this, I select page 1 appears, I select page 2 appears. Now I'm doing page 1. And these are the rate of diffusion in my experiment. The first one is 26.83. And I go back to page 2, 26.83. And I go back to page 1, 30.88 rate of diffusion in my second run. 30.88. Rate of diffusion for my latest run is 74.95. And I go back to page 2 and enter it here. Was it 74.95? Yes. 74.95. If I double click on this, it will open up options for this column, and name for y-axis was the rate of diffusion, and I type it in here, rate of diffusion. For a short name, rate of diffusion, I'll do this, and units is, I need microsiemens. So microsiemens per centimeter per second. That's the unit for rate. And I click done. And I go for the x-axis. Remember, x-axis was my independent variable, which is surface area to volume ratio, and short name for it, I'll do SA over V. And there's no units for this. So I click done. As you can see on the graph, on the bottom, the label for the x-axis, surface area to volume ratio, and on the left, label for the y-axis, rate of diffusion in microsiemens per centimeters per square. After I plot my data here, I really don't like the way that the software plots the data with empty circles. I want to change it and also the color. So I go ahead on one of the data entries on the graph and double click on it and go to options. Instead of empty circles, I will select filled solid circles, and then I will change the color to blue color. As you can see from the graph, there is a clear correlation between surface area to volume ratio and the rate of diffusion of these model cells. The higher the surface area to volume ratio, the higher the rate of diffusion. Smaller cells have a higher surface area to volume ratio, thus they have higher rates of diffusion, and they are more efficient cells in terms of transporting matter across membranes. If a cell is very large, the surface area will be lower and the rate of diffusion will be lower, making them less efficient cells in transporting matter across membranes. When comparing cells from organisms that are of different sizes, one can assume that large organisms will have larger cells. However, we know by experience that a liver cell, for example, from a human, from an elephant, from a mouse, are about the same size, aside from the natural variation. As we can see from the results of the experiment, smaller cells had higher rates of diffusion than larger cells. Collecting more samples would allow for better description and representation of data, as well as allowing for performing statistical analysis as needed.